This is George Kobakhidze from Baruch College, and this is for Bio 2010. This video is going to be on enzymes. But first, let us review proteins. And as you know, proteins are just a string of amino acids put together. So here's an amino acid bonded to another, bonded to another, and so on and so forth. You're not just a few, but many amino acids. The bonds that link these amino acids together, we call these peptide bonds. And these are an example of covalent bonds, which consist of two electrons being shared. To form these covalent bonds, we undergo a reaction called dehydration to form a peptide bond. And in a dehydration reaction, we lose a water molecule, lose an H2O. So let's review how proteins are made. So we're going to start off in the nucleus. And this is where a template is taken from DNA and it is used to create the RNA complements. This is called transcription. It's called transcription. Now the RNA goes to the next organelle called a ribosome. And this is where RNA is read, amino acids are brought, and protein is formed. So process from RNA to protein, we call this translation. Okay, and the last step is our newly formed chain of amino acids. Before it is called a final protein product, it is going to go through the organelle known as the Golgi apparatus. And here it's going to pick up a few attachments. So whatever it needs, based on whatever the protein is, pick up attachments. And now we can kind of call it the final protein product. Final protein product. Okay, now for some examples of proteins. The one we'll be dealing with today is called amylase. This is also an example of an enzyme, which we'll explain what those are in a sec. But amylase takes starch, which is a polysaccharide, and turns it into glucose. So this protein finds starch and catalyzes the reaction to glucose. Another example of a protein is hemoglobin. This is found in our blood cells, and they can carry and drop off oxygen. Carry and drop off oxygen. Carry and drop off oxygen. And the last example, which is of a different type, is actin and myosin. They are proteins, but they are structural proteins. So just like the name says, they are a part of a structure, and that structure is our muscle cells. So this is a structural protein, part of muscle cells. Okay, so now on to enzymes. Again, enzymes are just proteins, but these have a few attachments to help it do whatever it's supposed to do. First new definition is enzymes are also catalysts, and all that means is they speed up reactions. But it's even more than that. They allow reactions to happen, because without enzymes you might not even have a reaction, even if you wait as long as hundreds of years. So here's an example. Let's take this generic chemical equation. You have A plus B goes to C plus D. Let's say this one is without enzyme. The time for completion for this reaction could be on the order of years. So this kind of tells you how important enzymes are. It couldn't even ever happen. Now if we were to take this same reaction but give it the appropriate enzyme, so let's say this one is with enzyme, we could expect to see, take about 15 seconds or something. This just goes to show, we really need enzymes to speed up these reactions. And without them, we'd probably die. We would die. Okay. So one important thing about enzymes is that they do not get used up. They are regenerated at the end of the reaction. So here's our generic equation. I'm going to put enzyme here as a reactant, but you won't normally see enzyme written like this. It's usually above the arrow sign right here. So at the end of the reaction, we could see the enzyme is also here. So we start with enzyme, and we are also going to end with the enzyme. And then just note enzyme not usually written as reactant. It's usually above the arrow sign. So this usually above this symbol. Okay, so on a different level, we can say the enzymes lower the activation energy of a reaction. So let's diagram what that means exactly. So on the x-axis, we'll say reaction progress, not exactly time. And on the x-axis, we will say free energy. And we use the symbol G for that. And again, let's go back to our, friendly generic equation. So let's say in this case, the free energy level of our starting material is up here. And our products are going to have energy level down here. So we can call this, we can call these reactants, or you can call them starting materials. Starting material. And this we can call product, our products. For A plus B to become C plus D, it's going to have to acquire some energy, and it's going to have to surpass this barrier that I just drew in. So from here to here is the amount of energy we're going to need. We call this the activation energy. So this is the energy required for the reaction to occur. So in this diagram, we will say this is without the enzyme. So we will say, without enzyme. And without the enzyme, this reaction isn't very probable. There's not enough energy. We can't overcome this barrier. So we're going to say here that the reaction here is not probable. Okay, but now let's take a look at the same reaction, but with the enzyme added. take this picture, plop it in right there. Move this down. Okay. Erase this, and let's get this out of the way. Oops. Okay, but now this time we are going to have our enzyme. So with enzyme. And again, the x-axis is free energy. We will just say this, we'll just indicate that with a G for free energy. So with the enzyme, it lowers this activation energy, this barrier, which makes it easier for A to B to become C plus D. So if you notice here, we have less activation energy. So now we can say with the enzyme, with enzyme, the reaction is much more probable. Last thing to note here is the free energies of our starting material, so up here, and the free energy of our ending material, the products do not change. So let me write that. So the free energies of the reactants and products do not change. Only the activation energy changes. Okay, enzymes are both pH and temperature sensitive. So what that means is if the environment is too acidic, the enzyme is going to lose its shape. And a fancy way to say that is that it's going to denature, lose its 3D shape. It's going to denature. Once this happens, no matter how much denaturation, the enzyme will no longer function. It is completely useless. No longer function. Okay. The same applies if the environment might be too basic. So we will just draw an arrow following this path here. If it is too hot, if there's too much energy, so another way to say this is, we have too much energy in the system. This is going to break the bonds of the enzyme. The energy will break bonds on the, I guess we should say break bonds of the enzyme. And then this also leads to denaturation, and we can follow the same pathway here. So needless to say, enzymes have an optimal range. So enzymes have an optimal range, an optimal pH and temperature range where they work best. I will see you in the next video.